Welcome to this lecture on physiology of micturition. In this session, we will explore how urine is stored in the bladder and how the micturition reflex is controlled and how the nervous system regulates the act of urination. Understanding this process is essential because it ties together renal physiology, neurophysiology, and autonomic control. So we will take overlook of micturition. By definition, micturition refers to the process by which urine is expelled from the urinary bladder through the urethra. It involves two main phases, which is a storage and voiding phase. The kidneys produce urine which is transported by the ureters to the bladder. The bladder temporarily stores the urine until the appropriate time for voluntary release through the urethra. Then we will recall anatomy of urinary bladder once again. The urinary bladder has three major layers. The mucosa is lined by the transitional epithelium that stretches as the bladder fills. Secondly, the detrusor muscle is the thick, smooth muscle layer responsible for contraction during urination. At the bladder outlet, two sphincters regulate urinary flow. First of all, the internal urethral sphincter, situated at the bladder neck, controlled involuntarily by the autonomic nervous system, and the external urethral sphincter, made of skeletal muscle and which is under voluntary control. So this picture is showing the layers of bladder wall. As you can see, there is transitional epithelium, then there is lamina propria, then inside is submucosa, then there is detrusor muscle, and then last layer is the adventitia. From outside to inside, these layers are constitute the bladder wall. And on larger picture, you can see the detrusor muscle. You can see the ureteral opening situated on the interureteric ridge. You can see the trigone of the bladder. You can see the neck of the urinary bladder, and there is internal urethral sphincter situated at the bladder neck. Then what are the phases of micturition? The micturition cycle alternates between two main phases, that is storage and voiding phases. During storage phase, the sympathetic nervous system predominates, the detrusor muscle remains relaxed to allow the bladder to fill, and the internal sphincter stays contracted to prevent the leakage of urine. When voiding begins, parasympathetic activity takes over, causing the detrusor to contract and both sphincters to relax, allowing urine to flow out. As you can clearly see, this picture is showing phases of phases of storage and voiding. You can see for phase 1, filling and storage, where bladder neck is closed, external sphincter is contracted, detrusor muscle is relaxed, and contraction of detrusor is inhibited by parasympathetic control. In second phase, it is voiding phase, where detrusor muscle contracts, bladder neck descends and opens, and external sphincter and pelvic floor relaxes, and urine expelled through the urethra. And phase 3 is termination of voiding, when there is no urine in the bladder, then bladder neck became closed, external sphincter and pelvic floor contracts, residual urine above the level of external sphincter squeezed back into the bladder. So this picture is clearly showing the stages of micturition. Then there is neural control of micturition. Micturition is a highly coordinated event controlled by several neural centers. The sacral spinal cord houses the basic reflex arc for micturition. The pontine micturition center in the brain stem integrates input and ensures that bladder contraction and sphincter relaxation occur together. Finally, the cerebral cortex allows conscious control. This is why adults can delay urination voluntarily. Then pelvic nerves carry parasympathetic fibers which cause detrusor contraction. The hypogastric nerves provide sympathetic input for storage, and the pudendal nerve provides somatic control over the external sphincter. This picture is showing the neural control of micturition. As you can see, there is cerebral cortex, pontine micturition center. Then there is somatic nerve fibers of the pudendal nerve supplying the urethra. Then you can see the sensory supply, which sensory supply to the stretch receptors of the bladder. Then you can see the pelvic parasympathetic fibers here. Then what is the micturition reflex? As urine fills the bladder, stretch receptors in the bladder wall detect distension and send signals to the sacral spinal cord. This triggers a reflex loop that sends parasympathetic efferent impulses back to the bladder, causing the detrusor muscle to contract and the internal sphincter to relax. When this occurs, we perceive the conscious urge to void or urinate. If the timing is appropriate, voluntary relaxation of the external sphincter allows urination to proceed. This picture is showing the micturition reflex. Again, you can see here this picture is also showing micturition reflex is a coordinated reflex communicated by the higher brain centers. As you can see, the sympathetic and parasympathetic supply to the bladder. Voluntary, and this picture is also showing the micturition reflex pathway. Filling of urinary bladder lead to stimulation of stretch receptors in the bladder, which sends the efferent impulses passes via the pelvic nerve. Then sacral segments of the spinal cord are involved. Efferent impulses travels via pelvic nerves, causing contraction of detrusor muscle and relaxation of internal sphincter, causing flow of the urine into the urethra and stimulation of stretch receptors, which gives efferent impulses via pelvic nerve, leading to inhibition of pudendal nerve, which causes the relaxation of external sphincter and ultimately leads to the voiding of urine. Then voluntary and involuntary control of micturition. In infants, micturition is purely reflexive and involuntary process. As the nervous system matures, the cerebral cortex gains control, allowing voluntary suppression of the micturition reflex until it's socially appropriate to void. Damage to spinal or brain centers can disrupt this coordination, leading to conditions such as neurogenic bladder, urinary incontinence, or urinary retention. This picture is showing types of micturition. As you can see, voluntary reflex present in adults and involuntary reflex in neonates, and defect in maturation can lead to involuntary reflex in infants, and if there is maturation, it will lead to the voluntary reflex. This picture this So in the end, we will summarize all of our lecture. To summarize, micturition involves a finally balanced interaction between autonomic and somatic control. During the storage phase, sympathetic tone keeps the bladder relaxed and the sphincters closed. When voiding is initiated, parasympathetic activation contracts the detrusor and relaxes the sphincters, allowing urine to be expelled. This process illustrates the remarkable integration of neural and muscular physiology that maintains urinary continence and proper elimination. So, in the end, we will summarize everything into this one picture. Micturition is process by which urinary bladder empties when it becomes filled, involving two main steps: filling and storage. In first phase, bladder fills progressively until tension in its wall rises above a threshold level. This elicits the second step, which is the nervous reflex, which is also known as micturition reflex, that empties the bladder, or if this fails, at least causes a conscious desire to urinate. Although the micturition reflex is an autonomic spinal cord reflex, it can also be inhibited or facilitated by the centers in the cerebral cortex or brainstem. So thank you all of you for listening this lecture. We will see you in another lecture, and good luck until that time. Bye.